out, especially if you're in that Enterobacteriaceae family. But aside from INVIC, the accessory labs like phenylalanine, deaminase, DNase, and urease, they help you to just kind of narrow down even more. So INVIC is kind of a good, starting point, and then the coliform tests are also really helpful for narrowing down. But urease is nice because not too many organisms make urease. This one is a very straightforward test, which is also nice. And all we're going to do is we're just going to see if your organisms can break down urea. That's all we're going to look for. Urea is something that pops up in our bodies as we metabolize our proteins and amino acids. Urea is kind of like a waste product or a byproduct. And so that's how it comes about in our bodies. And then we know that there are some bacteria that can use it and break it apart, and use it as a source of nitrogen. And so we kind of use that to our advantage since we know there's not too many bacteria that can produce urease. We kind of use that as a way of filtering down a little bit, narrowing down our list of possibilities. I did mention, though, that urease, this experiment is the exact same experiment that they'll use for diagnosing people for ulcers. The only difference is on the actual test that they use for the biopsies, they wouldn't actually incubate it like how we did. So we incubated for 24 hours. That experiment, their urease test is a lot faster. They call it a rapid test, so they get the results a little bit faster, which is nice. But the concept is the same though. So in the disc that they use for diagnosing peptic ulcers, there's also urea in there, just like how our agar has urea in it. All right, and then what's really nice is when the urea is broken down, you only end up with two end products. Ammonia is the first one. Carbon dioxide gas is the other. Since there is no germ tube, we're not going to look for gas, we're just going to look for the ammonia. So we're going to look for a shift of pH, but instead of an acidic shift, we're going to look for a shift to a basic. We're going to look for a rise in pH. So that's all we're going to look for is the ammonia production. That'll be kind of a sign that urease is being made. All right, that Berge's manual actually does list in one of the columns, urease is one of the more popular experiments. Again, especially in that family, there's usually only some genuses that make urease. I want to give you a couple of examples. So I don't have all the examples, but these are kind of the main ones. Proteus is one, like that Proteus. We've worked with Proteus. Parabolas, we've worked with Proteus. I think Vulgaris. Both of those, which are linked to urinary tract infections, they're known to make urease. The other one is Klebsiella, which is linked to pneumonia. Probably, though, the more common organism that's linked to pneumonia is that Streptococcus pneumoniae that we looked for on the blood agar. Klebsiella, though, can cause pneumonia also. Helicobacter, I wanted to mention, has a different name, Helicobacter. Helicobacter is not in that Enterobacteriaceae family, but we have to mention it because this is the one that causes ulcers in people. And what happens is it lives in your stomach. So it actually hangs out in the stomach. And most forms of Helicobacter actually are harmless. Like they will not give you ulcers. In fact, a lot of us in this room might be carrying Helicobacter and you probably have no idea that you're even carrying it. But there's some people, unfortunately, pick up the ulcer-forming type of Helicobacter. And the reason why that one's so bad is because when it forms ulcers, it literally breaks down the lining of your stomach and small intestines. The way that this bacteria can live in your stomach is basically by urease. So we don't know of any bacteria that can live in your stomach. And this is the only one that we know that can. And the reason why is because it sits in your stomach stomach and it dumps out urease. And the urease converts the urea in your stomach into ammonia. And that ammonia basically kind of neutralizes the acid in your stomach. So the acid never kills Helicobacter because the acidity in your stomach is pretty low. It's like pH 2, which is really acidic. So that's basically the reason why it can live in your stomach. So we're going to talk about that more in lecture with Helicobacter. The way that you would pick it up is you would eat it because it moves through fecal matter, and pretty much everybody has been exposed to fecal matter at one point or another. But and then it just kind of hangs out in your stomach. And if you're unfortunate to be carrying the ulcer-forming strain, then it'll actually start breaking down the lining of your stomach, which is not good. So in order to clear it out, you actually have to be prescribed antibiotics in order to get rid of Helicobacter. Then those ulcers can heal over, have a chance to heal. Okay, so the last thing is just for your notes for the project. Keep in mind, I did tell you guys if you did the tablet to inoculate pretty heavily, but once in a while, sometimes the organisms, just like with fermentation, there's fast urease producers and slow urease producers. So keep that in mind. A lot of you guys will probably set it up though, and you'll get an obvious yes or no, kind of be done with it. Some of you though might get kind of a middle where it's like starting to change, and you'll notice, it looks like there's some ammonia that's starting to show up. If that happens to you, just don't throw it away right away. You can just put it back in the incubator for like an extra day or two, just to give it enough time for the color to develop, so you can be more confident about your results. So if that happens to you, you end up kind of in that slow category. Like it doesn't look negative, but it doesn't look like an obvious positive. You can always re-incubate just for a little bit longer. All right.
On our project or for today?
Oh, on your project, yeah. Today you guys are all going to get an obvious answer. Okay, so that little, we had one person do a Petri dish, right? So we did, we kind of divided it up. We had one person do a Petri dish, and then we had everybody else do a little tube, and we squirted a little bit of water, and there was like a little tablet in there. Either way, the ingredients are the same. You either did a urea agar plate, or you did the urea tablet. And the other reason why we had you guys do both is we want you to be comfortable with looking at both for the project, because a lot of times we'll have plates, but then we'll run out, and then he'll start putting the tablets out. Either way, though, the ingredients are the same. So in both of the agar and tablet, there is some glucose, there is some ketone, but this is not a fermentation test, so the glucose is not added for fermentation. Like these two ingredients are just for nutrients, strictly nutrients, just so they'll grow in the incubator. The main ingredient we want to see if they can use is urea. So the urea, that's our substrate, and we said it's kind of a byproduct of a breakdown of certain amino acids. So it shows up in our bodies, so that's why bacteria can use it. And then other than that, there is a little bit of buffer and phenol red, which we've used before. It's the pH indicator. And then other than that, it's just the agar. Agar was kind of, there is no agar in the tablet, so the agar would only be in the plate. But everything else is pretty much the same. So you guys are going to see if our organism can break down urea. We said though urea gets gets broken down into ammonia and carbon dioxide gas. Most organisms are pretty strong urease producers, but some do it a little bit slower. We're only going to look for this end product, ammonia. And what's really nice is it's a pH, we have a pH indicator in there that we've used already. So phenol red is the pH indicator. And that was the one that we did a while back where if it was acidic, it turns yellow. If it was neutral, it stayed red. And then what about if it was basic, what color did it turn? That was the deep or bright. So in this lab, we're actually going to look for bright pink, telling us the ammonia is being made. So we're going to look for the ammonia production. And then we said the organism that causes ulcers, so it sits in your stomach and it just dumps out urease. And it uses all the urea in your stomach, converts it into ammonia, and ammonia is perfect because ammonia kind of counterbalances the acidity in your stomach. The last thing that's also really nice is usually when organisms make ammonia, they make quite a bit of it, usually, enough to overcome the buffer. Because again, we're kind of adding the buffer to help stabilize the pH. The reason why we want it in there is because we have a little bit of a problem. There is peptone in there. There is peptone included in your slant. And we said at the beginning of the semester that when our organisms eat peptone, they also make ammonia, right? So when they metabolize peptone, they also make ammonia. So we have a little bit of a problem because ammonia is what we're trying to look for with the urease test. So we don't want these ammonias like conflicting with each other. So the buffer we kind of throw in there so that it'll kind of prevent any pH shift if they eat the peptone. So if they eat the peptone, it won't be enough ammonia to change the color of our media to pink. The only way we're going to get pink is if they actually make urease, because usually the amount of ammonia you get there is so much that it'll cause a shift in color. If they just eat the peptone though, our buffer will kind of kick in and we won't see any hot pink color develop. So you guys can grab your test tube.